Very good evening, everyone. So today's topic is about the lipid bilayer. So what is the lipid bilayer? Actually, all the human beings and all the life forms that exist on the earth, they're generally made up of cells, which is the tiniest particle that can be considered. And inside the cell, all the organelles, which generally differ from prokaryotes to the eukaryotic system. And all these cells, irrespective of whether the cells comes from prokaryotes or comes from eukaryotic system, we have a lipid bilayer around the cell. So why it excites me always? Because the lipid bilayer thickness is around 5 nanometer. And you know the voltage difference, the same we studied in physics in class 11th and 12th. And it is so much interesting in terms of cellular biology. So why it is important? Because all the instruments that have been installed in our houses, they generally works on 260 volts or it could be different if we talk about different countries, but it would be more or less similar. But if we talk about cellular biology, the cells that we have, so the thickness of lipid bilayer is very, very small. It is approximately 5 nanometers. So the difference between between the two surfaces means the outer surface, the external environment, and the inner side means the cytoplasm. So generally the difference arises because of different ions. The ion concentration gradient, the same happens with the physics because of different ions. So same happens inside the cells. So because of these differences, a voltage difference arises. And you wouldn't believe what happens in our cells. The difference could be in terms of lakh millions, lakh voltage. So if the thickness increases from nanometer to centimeter or meter, so it would be very, very high voltage, higher than the high voltage that we consider, the voltage that is sufficient to and that is sufficient for the train system, that is sufficient to start or to continue different types of engines, different types of systems. And so now come to the topic. So we have now it is made up of what? The cellular membrane is made up of generally two things. The one is protein and the another one is lipids. So if you are not familiar with these two, let me introduce the first one, which is lipids. So lipids are generally made up of carbons, and at the last you will have carboxylic group. So it is a very simple structure. So generally it is carbonic acids, not carbonic acid. Because of carboxylic acid, it is acid. Now coming to the another one, which is proteins. So basically proteins are made up of amino acids. And what are the amino acids? Amino acids are generally Zwitter ions. Why it is why it is considered as Zwitter ions? Or Zwitter ion generally is a condition where an amino acid generally has no charge. No, why it is no charge? Because of the equivalency of two charges that an amino acid consists. So amino and acid, amino group, so it has an amine, NH3 plus, and acid because it has carboxylic acid. So these two things which have amine group and carboxylic group. So these terms, because of these two, it is known as amphiteric molecule, and we have it can act as an acid, or other hand it can act as a base. Now coming to a different thing. Now coming about only about lipids. What are lipids? So we can see I took this picture from Albert, and it is a sixth edition. I think it is the seventh edition. It is the latest one. So what types of lipids we have? So basically we have glycerolipids, phospholipids, and sterols, which are abundant in the lipid bilayers. So let me introduce one by one. So the first one, which is glycerophospholipids. So what is the meaning of glycerophospholipids? Basically glycerol, so it consists of glycerol. So let me introduce what is glycerol. Glycerol is a very simple structure which has 3 carbons, and you will have hydroxyl group at each carbon atom. So we'll have CH2OH, CHOH, and then CH2OH. And if this glycerol is attached to a phosphate, and then a phosphate will be attached to a different moiety, and we can distinguish different types of phospholipids on the basis what type of head group is attached to it. Now the same structure we can see here, which is phosphoridyl ethanol amine, because glycerol is attached to two fatty acids because of two alcohols. So I already discussed with you, we have 3 alcohols, so 2 has been consumed in the bond formation with the fatty acids, while the third one is attached in the formation of bond with the phosphate group, and the group is attached to ethanol amine. So it would be phosphatidyl ethanol amine. The another one, which is phosphatidyl serine. So what is a serine? Serine is an amino acid, and if serine is attached, then it is known as phosphatidylserine. And ps, which is phosphatidylserine, it has a very important role in signaling. So if generally phosphatidylserine generally resides on the inner side of a cell. So if it is inside, if it is in the cytoplasm, it is okay. Your cell is functioning, all the things, all the things clear, all the equilibrium, all states are in equilibrium. But what happens because of certain translocators? Suppose you have different types of translocators, which is generally responsible for the transmission of one phospholipid from inner leaflet to the outer leaflet, or the opposite one. And we have different types of phospholipid translocators. Now, what happens if somehow phosphatidylserine generally shifted from the inner cell surface to the outer surface? Then the phosphatidylserine would be recognized, and it would be engulfed by macrophages. So it generally triggers cell death, and generally because of this, it is known as eat me signal. Now, what is a sphingomyelin, which we can see here is sphingolipids. What is a sphingomyelin? Basically, if it has a sphingosine molecule attached to it. What is a sphingosine? It is 2 amino 4 trans octadecan 1,3 diol. So generally it is not that much difficult. So if it has amine group which is attached to the second carbon, because it is 2 amino and 4 trans, and so basically you have to attach a double bond between 4 and 5th carbon, and octadecene because of 20 carbon atoms, and 1,3 diol. So you simply have to attach 2 alcohol. So the overall structure will be of 20 amino acids. Will be of octadecene. No, it would be of 18, because octa and decene. Now coming to the structure, we have a different type of molecule, which is the last one, which is about the cholesterol, the sterols. And the most common example of sterol is cholesterol. So what is a cholesterol? So cholesterol is what do you know about cholesterol? Many times we heard that cholesterol is bad. We must restrict the quantity of cholesterol in our body. So generally it is not that bad for us. It is generally required to maintain the integrity of cell membranes. So we have cholesterol, and so cholesterol is generally considered to be a steroid. What is the meaning of steroid? We generally heard steroid so many times in so many times. If I generally talks about the gym freaks, so the steroid is very much common. So steroid are generally compounds which have carbon four rings, which are four rings attached to it. So we can see here in the cholesterol we have four pyrrole rings which are generally attached, and we have a tail-like structure, and it has a hydroxyl group which is at a position 2. And so what happens when the because of this hydroxyl group, so this hydroxyl group is generally polar, where because of OH, which will be generally, it can remove H, and it would be a partially negative charge on oxygen, so it can make a bond with the head group. And so because I already told you, head group are generally formed in phospholipids, and phospholipids are the most abundant type of lipid molecules that are generally present in lipid bilayers. And so OH group will bind to the head group and the tail-like structure, because it is non-polar. All the carbon groups, if all the carbon groups are attached in a single line, then generally the alkyl chains are hydrophobic. So this chain will make bond with the or it we can see here in the figure, in figure 10.5. Yeah, we can see here how it attached. I already told you the hydroxyl group will make bond with the polar group, and non-hydroxyl group will make bond with the tail region, because the tail region is generally non-polar, because it is also made up of carbons, or you can say methyl groups, or it could be unsaturated, like because of unsaturation there is a king. Why king is important? Because it generally provides flexibility, and One more thing, why cholesterol is required in the membranes, because what cholesterol does, cholesterol generally generally has generally no impact on the flexibility, but but it generally causes, it generally compacts the structure of the membrane, so it generally prevents so many things. It could prevent the dangerous disasters that could happen. Now, coming to I already told about the non-porous, porous. So why is this important? So we have two examples. One is acetone, and one is acetone, and the another one which is 2-methyl propane. So because of acetone, so we have C double bond O and carbon and oxygen. Because of different electronegativity, we have a partial positive charge on carbon. We will have a partial negative charge on oxygen. So what would happen? This molecule, the acetone, will be able to make bond formation with the water, and that's why the acetone is generally, soluble in water. While on the other hand, 2-methyl propane generally can't make bonds with the water, and thus it is insoluble. Why these things are insoluble? We learned from the classes, we have been learning from our school classes. They're generally non-polar molecules. They will combine together and they will form precipitate. So why they are forming precipitate like structures? So basically, suppose we have water. So all the water molecules will be surrounding, and it is in the free form, because water is liquid. So generally, it is not so much compact structure. So what would happen? Because it is, as we can see here in the figure 10, in the figure 10.6 here. So in the figure 10.6, the water molecules will form a cage-like structure, and from distortion, we are shifting towards a more ordered structure, because all the water molecules will be combined together and will encircle, or it will be around the acetone 2-methyl propane molecule. And so it is shifting towards an ordered structure, and we can say that the entropy is decreasing. So it is not with the law. It is a the law which is very similar, which is very common, the law of spontaneity, or we can say the orderness of a reaction always increases. So here, because the entropy decreases, it couldn't be happen. So all the molecules will be together, and the water molecule will try to not make so many structures, so many these cage-like structures, to be surrounded by 2-methyl propane. So all these molecules will combine together, and a large shell structure will form. Now, this is the last, I think this is the last slide, which is about transmembrane alpha helices. And so what are these structures? Alpha helix, alpha helices are generally most abundant in the membrane? It is a question why it is not in other types of forms. So I can tell you one thing. I told you the proteins are made up of amino acids, and amino acids have C double bond or NH bond. So because of these two, so NH will make a bond with carbonate. So this there will be a bond formation. So and the peptide bond will form. So it would be C double bond or NH. But let me honest, this bond is not completely non-polar. No? It has a approximately 40% double bond character. So what happens, because this is a polar structure, and inside the membrane, we discussed, because of the tail region, it is hydrophobic, and the water is absent. So whenever we have polar compounds and water is absent, and so generally the compounds, or the molecule will tend to maximize the hydrogen bonds, and the hydrogen bonds can only be maximizing in terms of amino acids if it forms alpha helix, because alpha helix is a structure which has which generally has hydrogen bonds with n and n plus 4, and it is a structure which has highest number of hydrogen bonds. And so and the last one is about, yeah, this is also very interesting. So I told you about domains. So what are domains? Domains are very much specific regions, and within these regions, certain types of proteins and certain types of lipids, they are very much confined. What is the meaning of confined? Means they are fixed. They can't go into different areas. And the very important example is the GIT tract. So But we can see here we have basal surface, we have lateral surface, and we have apical surface. But what do you mean by apical surface? And so basically apical surface is the region which is directly in contact with the GIT lumen area, while the basal and laminar region is basal and laminar region is the region which is in contact with the blood. So what happens, generally, the transport of sugars, many sugars inside the cell is generally occurs with the import of sodium. So sodium is required. So how the sodium concentration remains constant because of these confined regions. How it's happening? Because we have sodium potassium pumps at the apical region, so it generally maintains the sodium and potassium concentrations, while we have the pumps which is sodium and pumps for the sodium and sodium and and other transporters. So in this way, the transport generally occurs from the extracellular side towards the from the lumen side to the extracellular side, and from extracellular to the blood. And if this confinedness we somehow disturb, and so the transport process wouldn't be occur. Thank you so much.